Okay, in the last section of 3.4, we learned about the way molecules move into and out of a cell by passive transport. As you may recall, there are two kinds of passive transport: diffusion, which works for small nonpolar molecules and causes molecules to go from high concentration to low concentration. Here's an example. When your red blood cells arrive at your lungs, they are depleted of oxygen, while your lungs are full of oxygen. Oxygen, a small nonpolar molecule, enters the red blood cells from the tissue in the lungs quickly until the red blood cells are isotonic for oxygen with your lung tissue. Now, the second kind of passive transport is facilitated diffusion. Molecules which are either large or polar will still move down their concentration gradient, but they need assistance crossing the membrane. The assistance they need is a transmembrane protein that makes a tunnel through the membrane. In both of these cases, energy is not needed. Now for this section, 3.5, we will learn how to move molecules against their concentration gradient. Now if you look at this picture here, it's more like trying to blow up a really full balloon. You can see on the outside there's only a few molecules, and yet we're trying to pump them across the membrane through this transmembrane protein to put even more inside where clearly there's already a lot. So it takes energy to blow up a balloon that's already mostly full than it does to try to blow into one that's empty. Okay. Now notice, like I said before, there's few molecules on the left and many on the right, but your cell needs to pump even more molecules inside the cell. This is going to take energy as well as a transmembrane protein, which you can see in blue here. We call this kind of transport active transport because it requires energy. Okay, so as we said, passive transport from 3.4 requires no energy from the cell. Everything moves down its concentration gradient. The only time you need active transport is when you are trying to pump a molecule against its concentration gradient. And it takes energy, which in your body takes the form of ATP. Now, why would your cell want to pump molecules against their concentration gradient? Okay, I'll give you an example that you will learn more about in the next chapter. In order to turn glucose into energy, that's that ATP molecule, for your body, this happens. As your body breaks down the glucose, it uses the energy to pump hydrogen ions into a very small space, causing an extreme concentration gradient. Because of the great tension of this difference in hydrogen ion concentration, the ions will try to get through the membrane to the other side to equalize the concentration. But there's only one place where the hydrogen ions can go through, a protein that uses the hydrogen ions going through to make ATP, which is the energy molecule of your body. So you can see that sometimes it's important to make an extreme concentration gradient difference, and that ends up doing some work that the cell needs done. Okay, we'll go into more detail on that next chapter. Now we're going to talk about two other ways that a material can get into and out of your cell. So let's start with the first one, endocytosis. Endo means in, kind of sounds like in, and that's true. It's material trying to get into the cell. Cytosis. Cyto means cell, so into the cell. All right, sometimes material that has to go into the cell is too large to fit through a transmembrane protein. Instead, what happens is the cell will pinch off a little bit of its membrane and make a vacuole, pulling the material into the cell. This takes energy, so it's considered active transport. Endocytosis is a process of taking material into the cell, and phagocytosis is a kind of endocytosis where the cell is actually going to eat or digest the material that comes in. Let me show you. Alright, here's a cell, something like an amoeba for example, that has found a little bit of food outside of itself and it senses it with its cell membrane. And what happens is that the cell membrane will pinch in, creating a food vacuole. Now normally the food vacuole is a globule all the way around, but we cut this in half so you can see the food is still inside. That way this cell has brought in this little bit of food and now it's going to digest it, which I will show you in a second. Okay, here's the food getting pinched in by the cell membrane. It pinches in a little bit of itself, so the cell itself gets a tiny bit smaller, and then you've got this nice food vacuole. That attracts a lysosome, which as you may remember is filled with digestive enzymes inside, and the food vacuole and the lysosome will fuse together, making a somewhat larger food vacuole, and then the digestive enzymes will digest the food, and eventually this vacuole will fuse with another membrane and let the energy from the digested food out into the cell. Because we're pinching in a food vacuole with food in it, we call it phagocytosis, because phag, P-H-A-G-O, means in Greek means eat or food. So phagocytosis refers specifically to pinching in food. But there can be other things that your cell brings into itself. And then finally, the last kind of active transport that requires energy is when you have a tiny vesicle that has some sort of molecule in it that needs to be expelled from the cell. What happens is that, and I don't have the picture before this, but you could imagine the picture before this, we've got a nice plasma membrane around a tiny vesicle that has whatever molecules inside. It gets close to the cell membrane, and the plasma membrane around the vesicle will fuse with the plasma membrane of the cell, making the cell slightly larger and naturally expelling the contents outside. Here's an example. Your pituitary gland may need to send some human growth hormone out to the rest of your body. The vesicle with the HGH in it will travel, usually on a cytoskeleton monorail, to the cell membrane, where the vesicle membrane fuses with the cell membrane, making the cell a tiny bit bigger and expelling the contents of the hormone out into the extracellular matrix, and it's now available for the rest of your body. This is called exocytosis, which of course sounds like exit. So exocytosis means fusing with the membrane and expelling material outside the cell. Endocytosis means bringing in, pinching off a bit of the cell membrane around something you want to bring in and bringing it inside the cell. Thank you for your attention.